Hello students, welcome to another chapter, Understanding Biology, chapter 23. In this talk, we are going to learn about prokaryotes. In this talk, you should be able to learn and explain the general characteristics and origin of prokaryotes, differentiate between prokaryotes and eukaryotes, and explain how archaea is different from bacteria and eukaryotes. So when the word prokaryotes is said, you kind of start thinking about bacteria. So which is accurate, bacteria are included in prokaryotes. So today we are going to learn about certain important characteristics of prokaryotes, their origin, how prokaryotes are divided into two groups, and how are they different from eukaryotes. So prokaryotes are the smallest and simplest of all the organisms. They are made with a single cell and they are too small to be seen through the naked eye. So you need a microscope to observe them. And when you think about the origin of prokaryotes, it is believed and the evidence are suggested that they are likely to be originated on Earth over 3.5 billion years ago. So how do we know this? So when it comes to the early events of the formation of Earth, it is very difficult for us to get an idea due to lack of fossil records. But we do have these tiny fossil microbes called microfossils that have formed in very old rocks. So when we analyze these microfossils, actually it resembles these present-day prokaryotes. So when you date these microfossils, they are dated about 3.5 billion years ago. This is what gives us an idea that these prokaryotes might have origined 3.5 billion years ago on Earth. Prokaryotes, therefore, are the most abundant forms of life on Earth. And it is estimated that about 1 to 10% of all the prokaryotes that are present on Earth is known and identified and characterized. That leaves about 90 to 99% of all the prokaryotes undescribed and uncharacterized. Nowadays, scientists have many molecular biological tools that allows them to understand, analyze, identify, and characterize these prokaryotes. Therefore, by doing this, in 1980s, a new type of prokaryote was identified. This actually led to divide prokaryotes into two groups, such as archaea and bacteria. So in 1980s, when archaea was first discovered based on the ribosomal RNA sequences, it was a very important discovery that allow us to divide prokaryotes into two unique groups such as bacteria and archaea, and it allow us to open our eyes to identify unknown of these many prokaryotes that have not characterized yet. We now found many unique characteristics in archaea. Even though they belong under prokaryotes, they are very unique. So today's talk, you will learn about what are the unique characteristics of archaea. So let's look at first some of the prokaryotic characteristics. So when you think about prokaryotes, they are fundamentally different from eukaryotes. Prokaryotes, which include both archaea and bacteria, we know that they are oldest structurally, they're the oldest life form, and they are structurally simple and most abundant forms of life. So when compare prokaryotes to eukaryotes, we know that there are certain characteristics that are not similar between prokaryotes and eukaryotes. So we are going to go one by one these characteristics and compare them with prokaryotes and eukaryotes and see how different they are. So prokaryotes differ from eukaryotes in many features. The first one we are going to look at is unicellularity. So when you think about prokaryotic cells, With a few exceptions, prokaryotes are single-celled. In some cases, these individual cells can gather together to make filaments. These filamental structures, even though they actually have many cells that are aligned to make filaments, they actually still remain their single cellularity, individuality. So that means that you have a structure, filamental structure, but each cell actually keeping their individuality. So that doesn't mean that they are a multicellular organism. And also most bacteria in their natural environment are able to form these things called biofilms. Biofilms are actually complex structures that made out with different species. For an example, in your teeth having plaque, plaque is actually a biofilm that contain many different bacterial organisms coming together, forming this hardy structure that protect themselves and allow them to adhere to your teeth. So another character that we are going to look at is cell size. But before that, I had to mention that when compared to prokaryotes, you know most eukaryotes are actually structurally complex and most of them are multicellular. But there are many, actually many eukaryotes that are single cellular as well. So let's move on to the next character. We are going to look at cell size. Prokaryotic cells vary greatly in size if you think about it. There are very large bacteria that have discovered so far. The largest bacteria that is currently known is called Thymargarita nimbiensis, and it is actually visible to our naked eye. But usually most prokaryotes are only about 1 micrometer in diameter and they're very small. But when compared to prokaryotes to eukaryotes, many eukaryotic cells are well over 10 times bigger compared to prokaryotic cells. So the next character we are looking at is chromosomes. So the chromosome is a single cell when compared to eukaryotes. Bacteria and prokaryotic chromosomes are actually single circular double-stranded DNA. They have one big chromosome in the center of their cytoplasm, and it is usually arranged into a nucleoid which contain proteins and some histone proteins and the DNA together in this region. Compared to prokaryotes, eukaryotes have a membrane-bound nucleus and linear chromosomes that made out of both nucleic acids and histone proteins. And it is important to notice here that eukaryotes actually contain a prominent nucleus, a membrane-bound nucleus, which prokaryotes does not have. Their chromosomes is actually in the cytoplasm, and sometimes prokaryotes can have 2 chromosomes, but usually it's a single chromosome that is circular in nature. And many prokaryotes contain these plasmids. Plasmids are single circular double-stranded DNA that are smaller than the chromosome, and they sometimes contain very important characteristic features that are advantageous for prokaryotes. So the next character we are going to look at is cell division, and when compared to eukaryotes, prokaryotes have this cell division method called binary fission, which is a form of an asexual reproduction. The true sexual reproduction occurs only in eukaryotes, and that involves production of haploid gametes that fuse to form a diploid zygote. This process actually involves mitosis, meiosis, formation of spindle fiber. So these are actually, you already know some of these through your Intro to Bio 1 lecture. So remember, so only the true sexual reproduction can happen in eukaryotes. Prokaryotes does not have sexual reproduction. They have this method called binary fission, which is a type of asexual reproduction. And they do have the ability to exchange genetic material through this method called horizontal gene transfer, which we'll talk about in a little bit. So the next characteristic we are going to talk about is genetic recombination. And I mentioned that even though bacteria have asexual reproduction, they have the ability to certain times to exchange genetic material with a different bacteria. This actually occurs through horizontal gene transfer. It is not a form of reproduction, but a form of exchanging genetic material between bacterial species or different bacteria. This allows certain genetic diversity for these bacteria. Another character we will look at is internal compartmentalization. Cytoplasm, if you look at prokaryotes, their cytoplasm is actually does not contain any internal compartments or membrane-bound organelles. But in comparison, our eukaryotic cells contain membrane-bound organelles and internal compartments and endomembrane system. This is actually one of the unique characteristics of eukaryotes compared to prokaryotes. But both eukaryotes and prokaryotes contain ribosomes. So even though these ribosomes are structurally very significantly different between eukaryotes and prokaryotes, but both organisms, eukaryotes and prokaryotes, contain ribosomes. And it is very important to notice that ribosomes are generally described as organelles, but they are not bounded by a membrane. So since prokaryotes have ribosomes, you cannot tell that they have membrane-bound organelles because ribosomes are not membrane-bounded organelle. Next character we are going to look at is flagella. So prokaryotic flagella are structurally and functionally different from eukaryotic flagella. We all know that flagella are actually involving in locomotion or used to make the food to bring it near to the organism for eating. So when you look at prokaryotic flagella, they are composed of a single fiber of protein flagellin. The function of bacterial flagella, they actually function as rigid propeller. Unlikely to compare to eukaryotic flagella, they are complex and have 9+2 structure of microtubules, and their motion or their function of the flagella is like a whip-like motion. The last character we are going to look at is metabolic diversity. So you all know metabolism actually describe how an organism produces energy. So we are going to talk about two different metabolisms. One is photosynthesis, the next one is chemolithotropy. When you look at eukaryote only display certain photosynthesis that is called oxygenic photosynthesis that actually release oxygen. But when you look at bacteria, especially photosynthetic bacteria, they can have both the oxygenic and anoxygenic photosynthesis. Oxygenic photosynthesis release oxygen, while anoxygenic photosynthesis actually does not release oxygen. And we also, prokaryotes such as bacteria, can have another type of metabolism called chemolithotropy. In chemolithotropy, they can produce carbohydrates using energy stored in chemical bonds of inorganic molecules. And eukaryotes does not have this type of chemolithotropic metabolism. So that is another difference between eukaryotes and prokaryotes. Now that we're done talking about unique characteristics of prokaryotes, let's look at how unique Archaea is compared to bacteria and eukaryotes. So even though as prokaryotes, Archaea and bacteria share some characteristic features such as non-membrane bound organelles, unicellularity, and etc. But with current techniques and tools available, we found that archaea are unique compared to both bacteria and eukarya. By finding archaea in 1980s allow us to develop these three domain systems such as bacteria, archaea and eukarya domains. But further understanding and analyzing archaea, we actually found out many differences of archaea, and we are going to discuss about these differences in a little bit. But in 1980s, what actually make archaea to break from prokaryotes is differences in 16S ribosomal RNA sequences. And now we know much more unique features of archaea, both in structurally and metabolically. Now we are going to look at what are these unique characteristics of archaea. So let's look into archaeal cell structure first, which is the most obvious structural difference in archaea. So when you think about cell structure, one of the main things comes to our mind is the plasma membrane. So plasma membrane, all prokaryotes and eukaryotes have a plasma membrane. And in bacteria and eukaryotes, the lipids in the plasma membrane, phospholipid bilayer, are hydrophobic fatty acids, fatty acid tails that are actually ester bound to glycerol 3-phosphate. So here both bacteria and eukaryotic lipids are actually similar. They have glycerol bounded to this hydrophobic fatty acid tail through these ester bonds. Now, when you compare archaeal membrane lipids, archaeal membrane lipids are different. They are non-polar hydrocarbons, and these hydrocarbons are called isoprenoids. And they contain a glycerol skeleton with ether linkages. They are not ester, they are ether linkages. Here is their archaeal isoprenol lipid here. And that is very unique. That is actually different from both bacterial and eukaryotic lipids. So these lipids are actually these archaeal lipids. They can be branched or have rings. And another unique thing about archaeal lipid is that they may be organized into tetraethers. They can be actually used to form monolayers. And this unique lipid composition allow archaea to have membrane stability in extreme environments. So it allows these extremophiles to withstand high temperatures. So now you know how the membrane structure or the membrane lipids are different between archaea and bacteria, and also in comparison to eukarya. So another distinct structural character of archaea we are going to look at is the cell wall structure. So archaeal cell wall are unique compared to bacterial cell walls. So when you look at prokaryotes, they typically have cell walls. So in cell walls, the bacteria actually have this thing called peptidoglycans in their cell walls. But archaea does not contain peptidoglycan. They actually contain this pseudomurine, which is similar to peptidoglycan. That is one of a unique difference between archaea and bacteria. So we have another unique characteristics that separate archaea and bacteria is when it comes to this DNA replication. Most archaea have a single origin of replication, similar to bacteria, where their DNA replication is starting. But when you look at the proteins that initiate the DNA replication, it is very similar to eukaryotes. Same scenario is actually true for gene expression. So similar to bacteria, archaea shows coupled transcription and translation. The reason for this is because their nucleus, they don't have a nucleus. Therefore, their transcription and translation both happen in the same time in the cytoplasm. But when you look at archaea, the enzymatic machinery is very similar to eukaryotes. So Archaea have more than one RNA polymerase. Their enzymes closely resemble the eukaryotic RNA polymerase, and their translation machinery is more similar to that of eukaryotes. So that is all what we are going to talk for this topic. So in summary, prokaryotes are the oldest life form on Earth, which is now we divided into archaea and bacteria. Prokaryotic characteristics are shared by both. There are some prokaryotic characteristics that shared by both archaea and bacteria. And when you look at archaea, there are unique characteristics compared to bacteria and eukaryotes. So we will see you again in another lecture, and thank you very much.